All right, so good morning, everyone. Welcome back to KDA 2022. Yesterday, I think, was a very successful day. The PFDD sessions, the briefings by Dr. Grunstein, Dr. Al-Qahtani were just awesome and really set the stage for a successful conference. So we're going to continue with patient presentations this morning. We have three really good presentations lined up. And then after lunch, and also a roundtable with clinicians that will start after the first break. And then after lunch, we're going to go into the research portion of the conference. Now, for our first speaker, Ed Meyertholen really needs no introduction for people who've been associated with KDA for more than a year. For people that have joined just in the last few weeks or months, or people who are at their first conference or participating in their first session on Zoom, I'll just say that Ed is a mainstay of the KD community and of the KDA. He's served in many roles in the past. He is now our patient liaison to the Scientific Review Board, which is the group of senior researchers and doctors that review the grant proposals that we receive every year. Ed, as you may have heard yesterday, has a fascinating personal story. He started running marathons after he was diagnosed with Kennedy's disease. And he's still running today, as he says, almost as fast as his 4-year-old grandson. So that's all good news. More importantly and more relevant for today, Ed has a PhD in biology, is a KD patient, and really knows whereof he speaks. And so as we traditionally do in these conferences, we're going to start out with Ed giving a basic introduction to Kennedy's disease, a presentation that we've come to know as KD-101. So Ed? Welcome.
Okay. First of all, I'm just going to say this is a picture from 1960. I'm that old. I'm the middle one, and those are my two cousins. And little did we know that all three of us were going to end up having Kennedy's disease, or we haven't even at that point. And so it's always kind of a reminder to me what it used to be. So what I really try to do is I do not try to review the research in Kennedy's disease. You're going to get that from all the people who really do it. What you're getting from me is basically a 3-hour biology class, 3-credit-hour biology class in 40 minutes. So I'm going to go very fast and only do the highlights. And the other thing that you realize is that because it's very general, I make generalizations that there might be exceptions to. So just realize that, there's a lot of truthiness in what I say, but there's also sometimes exceptions. And I try not to give too many of my opinions, but I fail at that. So let's see. All right. So in 1968, William Kennedy published a paper that described Kennedy's disease. It's where the name comes from. Later, someone noticed that in 1897, there was a Japanese paper that seemed to describe Kennedy's disease. It's probably not sure if that it was, but it seemed like. And so it makes you wonder whether or not it really should be Kawahara disease instead of Kennedy's disease. The good news, of course, it's still a K, so the KDA stays just fine. But with Dr. Kennedy's paper, he talks about muscular atrophy. Now, yesterday, Chris Grunstein talked about all the different symptoms you can have, so I'm not going to go over much of those at all, other than two things. One is that there's a muscular atrophy, and muscular atrophy is your muscle cells are shrinking. So here is a cell that's not shrinking, nice and strong. Here are ones that are shrunk. Smaller cells, less strength, more weakness. And this is what's happening to us. And basically, despite all the other different symptoms that we do and do not have, we all are doing that. The second thing is a loss of certain neurons. And this is a picture of the spinal cord over in the, I think this works, right? Oh, right down in here in this region, you can see all these big dark spots. Those are what are called motor neurons. This is from basically, I don't want to use the word normal, but it's a normal person, someone who does not have KD. If you look at the KD in the same region, you see those spots are missing. So we are losing neurons. So we have both muscles degenerating or at least atrophying, and we're losing neurons, nerve cells. So it's a real neuromuscular disease. Now, what I'm trying to do is kind of give you possibilities of why this is happening. Now, to get an idea, whenever you have a disease, if you want to treat it, you have to know what causes it. And, if it was a bacterial infection, you would look for an antibiotic. If you had a virus, you look for a vaccine. Well, back to Dr. Kennedy's paper, he noticed that the sex linked. He says it's recessive. I think I could argue that it's not, but it's unimportant to us. The sex linked is important because what that means is that it's a genetic disease. Then we already know that. But if you say that to a scientist, that has a different meaning. We think of it as, we can pass it off to our kid, but a scientist says, well, you know what the cause of it? The cause of it is a protein. And now we have another disconnect between scientists and the rest of us. When we say protein, we're thinking meat, or if you're a vegetarian, tofu. That's not the protein you want to be thinking of. When I used to teach, I used to use an analogy of a cell as being like a house. And in the house are all these instruments and tables and things on the wall and ovens, etc. Those are all machines. And a protein are the machines of cells. Okay, so you don't think of it as food. You think of these are things that the cell creates, they make, and they do things in the cell. And obviously, if a protein doesn't work, that's not good. That means you're not doing something that the cell should be able to do. Now, what makes up a protein? Proteins are made up of amino acids. There are 20 different amino acids, and a protein is a linear string of amino acids joined together. What makes each protein different from each other is the sequence of amino acids. And again, back to my teaching days, an analogy is like letters and words. With 26 letters, you can make an infinite number of words. And what determines what a word is the sequence of letters. The difference between these two analogies is size. Other than if you speak German, a long word is maybe 10, 12 letters. If that was a protein of 10 and 12 amino acids, that would be extremely small. Proteins can be easily hundreds of amino acids, and some are 1,000 amino acids long. And the sequence that determines that is precise. You need to have that precise sequence to get that protein. So when you think of a sequence of proteins, I basically look at a little kid's toy, think of each letter as an amino acid. It's a linear sequence, but notice it's able to turn and fold. And proteins can fold in amazing ways. So these are real pictures of what the proteins look like. And these are just 12. Partially I pick it because they're very large, but the point is they're basically still, even though they have this dramatic three-dimensional structure, they're still built from linear strings. So it's kind of like a snake that wraps up. Now, it's important to realize that the three-dimensional structure is critical for the protein to function. If a protein is supposed to fold in a certain way and it doesn't fold that way, it will no longer work. So there is a critical relationship between the structure and the function. Now, what causes it to fold that way primarily is its amino acid sequence. So there is a relationship between the amino acid sequence, the folding, and the function. Okay. Now, at this point, my students would be asking exactly this question. What's on the test? And I've always wanted to give a test here. I haven't done it, but someday I will. Well, proteins are the doers of a cell, and the cell requires them to work in order to do what the cell's supposed to do. They're composed of long linear chains of amino acids, each protein having a unique sequence of amino acids. The sequence of amino acids causes the protein to fold into a unique shape, and that shape determines the function. And probably the crux of everything, if a protein should not fold correctly, it's dysfunctional, and it could cause serious problems to the cell. So realize if the protein doesn't have the right structure, it doesn't work. And that can be very bad. Now, how do you make a protein? We already know one thing you need amino acids. Get amino acids, either your cells can make them, but there's about 6 or 7 or 8, I should know the number, but I don't, that you have to get from food, which is why we had the proteins we ate from before, to get those amino acids. You need something that makes the protein, and it's called the ribosome. It's a structure in the cell. And finally, you actually, to make a specific protein, you need to know the amino acid sequence. And so that leads to questions, how does a cell know what the sequence is? If we go back to the idea of words, how do you know how to spell a word? And the answer is, well, there's a dictionary. So your question should really be, what's the dictionary for proteins? How does a cell know what, 1,000 amino acids are? How do you know the sequence? And the answer is, it's in your genes. That's what your genes are. They're not traits. They are instructions on how to make a protein. So now that, of course, gives you another question. What does a gene made up of? Probably know that's made up of DNA. That's not very helpful. What's DNA made up? Excuse me? It's made up of 4 bases. And like proteins, it's a linear string of these 4 nucleotides. I said amino acids, but they're nucleotides. A, C, T, or U. There are differences, but to us, T and U are basically the same thing. So they are long strings, and they're just of these 4 nucleotides. Each gene has a specific sequence of nucleotides. And coded into that sequence is the amino acid sequence for a protein. For example, if you wanted to make a protein that you've all heard about, let's say insulin, there exists in your DNA a stretch that has the nucleotides that coded in there is the amino acid sequence to make insulin. So it's pretty amazing. Now, the code, so here's a small section of DNA, the UUU, GUC, and for those who are technical, I know the U is not in DNA, but it's fine. And we find that the code is actually based upon groupings of 3 nucleotides. So if we break them up, instead of a long string, you see it's a string of 3 sets of UUU, GUC, AAA, UGA. These are known as codons. One of the few times a biologist has named something that makes sense. Now, it turns out that if you know the genetic code, you can just, and it's, there's actually tables. I used to show tables, but I'm not today. UUU stands for the amino acid phenylalanine, GUC, valine, AAA, lysine, and UGA means stop, that the protein's over. There are 64 different possible codons, and every codon either codes for an amino acid or codes for a stop. And the most amazing thing to me is that it's universal. It's a plant, a fungus, an animal, a human. The genetic code is the same. And that's important to us because you know that there are animal models of Kennedy's disease. And what have they done? They've taken a human gene and put it into a mouse. And the mouse, because the genetic code is the same, can make the human protein. So, it's really quite amazing. Now, let's talk a little bit about how you actually make the protein. So here is my typical cell. You know about the nucleus, that's where the DNA is. And the DNA contains millions of nucleotides that are part of maybe hundreds or thousands of genes on one piece of DNA. So DNA is huge. And it cannot leave the nucleus because it's so big. Now, out here in the cytoplasm are the ribosomes. And remember, the ribosomes are where we make the protein. So now we have a small problem. The ribosome is too big to get into the nucleus. The DNA is too big to leave, excuse me. And if you want to make a protein, you need the information the DNA has. So how do the two meet? And again, I go back to my teaching. I used to pretend, suppose I tell my students there's a book in the library that's on reserve. It's 5,000 pages long. you have to read pages 6 through 8. And you don't want to do it on reserve. What do you do if you can't take the book out? You make a copy. You make a Xerox copy, and that you can take out. And that's exactly what happens when you make a protein. You make a copy of the piece of DNA that codes the protein you care about. It's in the form of another molecule called RNA. That's much shorter, can read. It goes to the ribosome. The ribosome then reads the code and makes the protein. To look at it kind of in a different picture that I stole, you can see that the nucleus has a DNA and you make RNA from it. That's a copy. It's not a chemical reaction. That process where you make the RNA is called transcription. Okay. The RNA then leaves, goes to the ribosomes, and you make the protein. It reads the code and knows which amino acids to put in. That's called translation. It's the first time of real jargon I think we really talked about. So it's a two-step process. And again, that's universal. The information flow to make a protein is always from DNA to RNA to protein, and there are no exceptions. There is no protein your cells make that isn't from a gene. And you can only make a protein based on the genes you have. So if you have the wrong sequence of nucleotides, you will make the wrong sequence of amino acids. And that can have catastrophic effects. Catastrophic is a bit. So back to the test, what do you need to know? DNA is composed of nucleotides. The sequence determines the amino acid sequence of proteins. Proteins remain in a two-step process: transcription, which is DNA and RNA; translation, RNA and protein. The specific piece of DNA that codes for a specific protein is what we call a gene. And it's that what you inherit from your parents. You inherit instructions, really, not traits. And finally, again, the crux. If the sequence of nucleotides in DNA is altered, we call that a mutation, then the amino acid sequence of the protein may be altered, and the protein may not fold correctly and therefore not work correctly. And now, hopefully, you see the relationship between a genetic disease and a protein. The genetic disease is a mutation in a gene that codes for a protein, and that protein evidently does not work correctly and causes the disease. So I would be making the class do this next part, but I'll do it for you. Since KD is a result of single gene mutation, and each gene contains the instructions to build a specific protein, then there must be a specific protein that's mutated that causes KD. And 31 years ago, Alice Bonner from Kurt Fishback's lab identified the androgen receptor gene as the one that is mutated and causes Kennedy disease. Once again, I want to thank them because they named the gene very logically. The androgen receptor gene is the gene that tells you how to make the protein androgen receptor. So nice. Now, what's the mutation? And this is where it gets actually very interesting. This is the nucleotide sequence of the androgen receptor. And in red, which you can't see, but I'll enlarge it, is a special region that goes CAG, 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 etc. There are 20 of those in this one. But it turns out that 20 is just the number I picked. Different people have different numbers of CAGs in that region. It can be as low as, I want to say, 16 or so. It might even be lower than that. It can be as high into the 60s. Okay. The reason it matters is because what Al discovered was that if you have more than about 35 repeats, you get Kennedy's disease. If you have less than 35, you don't. And this had never been seen before in a type of mutation. Very weird. It's called a CAG repeat. And of course, what do we all know? We know how many CAGs we have. I'm 46. Right? And so therefore, it sounds good until I realize it's above 35. So not quite that good. Now, CAG, of course, is a codon. That means it codes for an amino acid in the protein. So if you look at the amino acid sequence of the protein, once again in red, there is, now I know you're going to say Q, what's that? When the amino acid CAG codes for is called glutamine. And what else could glutamine be if it wasn't a Q as an abbreviation? That was a joke. I have no idea why it's Q. But because of that, it's called a poly-Q disease. And I will have 46 glutamines or Qs in that region of the protein. So what happens is that the more CAG repeats you have, the longer that poly-Q region is. And that evidently affects the folding of the protein and affects how it works. Now, what then became even more interesting is that over the next few years, 8 other diseases were found to be caused by CAG repeats. All of them in different genes, but they all have one thing in common, 2 things in common. They affect, I guess it's one, they kill nerve cells. And so there must be something about those repeats that nerve cells don't like. But they do different cells, so there must be something about the function of the protein that's important as well, because different proteins affect different cells. Now, the answer is we really don't know about, but it's pretty amazing. Now, how does the mutant AR cause KD? Now, I'm going to give you some possibilities. They could be right, they could be wrong, but at least it makes sense. But the first thing you have to know is a little bit about the androgen receptor. So I have a little cartoon about the androgen receptor. But we have to use the jargon. So I want to make sure that you understand what the things are that we're talking about. So we already know about some of this jargon. Transcription is just another name for RNA synthesis. Translation, protein synthesis. Protein modifications. This is one that you wouldn't have expected. It turns out that proteins, the cells can modify proteins by adding small molecules to them. And what this does is it affects the way it folds, and thus it affects the way it works. And so I look at it as these are like little switches. And the cells can turn them on and off by putting on and taking off these modifications. Over the next 2 days, you will probably hear a lot of words that are modifications, and you may not know that they're modifications. So I'm going to tell you how you can tell. Some possible modifications are called methylation. This is when you add a methyl group. Now, who knows what a methyl group is, right? But nonetheless, or phosphorylation, or acetylation. What do they all have in common? They all rhyme with modification. So if you hear someone say, oh, there is this ubiquitination, you're going to say, I bet you that's a modification of the protein. And you'd be right. So you may not, it doesn't really matter to us whether it's methylation or phosphorylation. It does to the researchers, but it doesn't. Just know that it's a modification and you're playing with the way the protein works. And you'll see why we care. There's something called a proteasome. This is a very large protein complex. Actually, you saw it earlier when I showed you the 3-dimensional proteins, you just didn't know it. It's huge. But what it really is a protein shredder. Proteins don't last forever. You got to get rid of them. You want to recycle the amino acids. You take them, put them in the proteasome, and out the other end comes, and it's a little bit wrong, amino acids that you can reuse. It's a little bit more complicated, but pretty much that's what it is. It shreds them up. So it's the way you get rid of proteins you don't want anymore. There's also something called autophagy. There's another way of getting rid of proteins. So think of it maybe as like, you have 2 streams in your trash. You have trash, that goes to the proteasome. You have recycling, that goes to the autophagy. Again, you'll see why we care as we get along in the talk. Then there are things called co-activators. Now, this isn't going to make sense at all yet, but these are helpers of the androgen receptor. And then it'll make more sense when we show what the androgen receptor does. And finally, there are chaperone proteins. Again, this is one time when they actually named them very nice. These are proteins that watch over other proteins to make sure they don't get into trouble. I kind of think of them as molds. So they hold the protein into a specific shape so that it doesn't change the shape, gets us away, and keeps them out of trouble. And again, you'll see where that comes from, I hope. So, in my cartoon, we have androgen receptors. We have androgens, and just so it's clear, androgens are male sexual hormones. You know, the most common one is testosterone. The next one is dihydrotestosterone. So when I say androgen, you can think testosterone. And that would be just fine. You have the modifications. I don't care if it's phosphorylation or methylation. They're all green to me. We have chaperone proteins, which of course are labeled HSP, which makes no sense. We have the co-activators, and we have DNA. And these are all going to be the parts in our play about the androgen receptor. So let's look at someone who has fewer than 35, so therefore they do not have KD. They have in the cytoplasm the androgen receptor, right here. Attached to it are the chaperone proteins. So that protein is being basically locked up and kept away from doing anything. This protein basically does nothing in this state. It floats around, in the cytoplasm, after about 1 or 2 or 3 weeks, the cell says, it's too old, we got to get rid of you. So it sends them to the proteasome and it replaces it. Now, mind you, it's not just one, you have many copies of this. So you're constantly destroying and making new androgen receptors. But the key is when there's no androgen, no testosterone, nothing's happening. So to some extent, this is what it's basically like in females. They have some testosterone, let's assume they have none. The androgen receptor doesn't really do anything. It just floats around. Now, what happens if you bring in the androgen? Now all hell breaks loose. And there are a number of big changes that occur. I'm going to do them in sequence, but this isn't the sequence in which they occur. It's not even where they occur. It's not important to us, but you get a number of changes. First thing is you get rid of the chaperone. So now you're free to get out. You get modified. And the types of modifications you get vary depending on what cell type you are. So it's possible that a nerve cell and a muscle cell and a liver cell are going to modify it slightly differently. And we'll see why that matters. They join, of course, this is what the chaperone are trying to stop, right? They didn't want 2 of them to get together, but they do. So now they're in dimer. And now the biggest change is they now go into the nucleus. In the nucleus, they're then attracted to those co-activators. And the co-activators bind. And once again, the specific co-activators differs from one cell type to another. So even though we have the same androgen receptor, we're going to see the activity of the androgen receptor is going to be slightly different in one cell compared to another cell because they have different co-activators and possibly different modifications. Hopefully that makes sense. And then what they do is it then binds to DNA and acts as something called a transcription factor. And this is a protein, a transcription factor, and that's what the AR is going to do, that turns on transcription. Now, again, if you're in my class, I say, well, what's transcription? The first step in, and you're going to say protein synthesis. So you're going to start making new proteins. Which proteins you make depends on where you bind on the DNA. And that depends on which modifications or co-activators you have. So a nerve cell will make one set of proteins. A muscle cell might make another set of proteins. So even though we're using the same system, we're able to get different reactions in different cell types, which is pretty cool. Now, once again, they again only last for a couple of weeks, and then go back out, go to the proteasome, and we're all done. And that's essentially what happens in males when they have, after puberty, when they have testosterone. Now, women have some testosterone and some androgens, so they have some of this happening. It's just a much smaller amount. Now, what if you have more than 35 repeats? Well, when we're on this side, it works exactly the same. If there's no androgen around, it goes to the proteasome and it gets destroyed. No problem. Nothing happens. So that's why, since women have most of theirs in this case are carriers, they're unlikely to have the major symptoms of the weakness that you see in the men. But as we go on, you'll see because they have some, it's going to be somewhat like the men. So it's kind of, you'll leave it at that. So what about on this side? Well, it's possible that having the change in the CAG repeat affects how it's modified and affects which co-activator it's binding. So in other words, remember, since that determines which genes you turn on, it could be that the people who have more than 35 repeats might turn on slightly different set of genes. And it could be because you're making different proteins, the cell may not like that. It might atrophy if your muscle, it might die if it's a nerve. One possible mechanism is that you have altered activity of the androgen receptor because you turn on different genes. I'm going to call this the transcription hypothesis. It could be wrong. It could be something I made up, but I like it. It makes sense. But that's not the only hypothesis. It also turns out that when you try to recycle them, the proteasome can't break it down. Can't break down the ones that hadn't gone to the nucleus. And you can actually see results of that in nerve cells on the left side. That's the nucleus of a nerve cell. This is this big black thing in it. When you look up close, it's a big smash. And what it is, it has little has parts of proteins in it. And part of the proteins in it are parts of the androgen receptor. There's other proteins, but that's one of the ones that you can find in there. These are only seen in people who have KD. It does not occur in people who don't have KD. So that's that's kind of the idea that you're not breaking down the protein well enough, and some of the parts remain. And it could be that those extra parts of the cell build up, kind of like when I was a kid. There was a trash strike in New York City, and the pictures had trash blocking streets. And so the city actually had to shut down in certain parts because of all this trash in the way. In the same way, it could mean that this extra trash in the cell causes it to dysfunction and not work. This I would call the trash hypothesis. Again, I made these up. But I mean, the point is that's a possibility. And there could be other ones not affected. Now, based on those, how can you go about trying to treat KD? How are you trying to do it so the person who has the 35 repeats or more doesn't have problems with whatever, either the transcription hypothesis or the trash hypothesis? Okay, so one way is by reducing or blocking androgens. Realize if the androgen receptor stays out in the cytoplasm, nothing bad happens. So what if you take away all the androgens? Or what if you add something that blocks the receptor on the androgen receptor, where the androgen binds? Then they can never bind, it will never go inside the nucleus, and you'll never have a problem. If you do this with mice, it seems to work. It doesn't seem to work quite as well in humans. The two back in 2006, 2008, there were two clinical trials, both of which were designed around changing the levels of androgen receptor. And the results were, I guess you can say, mixed. It certainly wasn't a resounding success. But that is one way that would work. Okay. The second one is the modulation of AR function. So this is attacking the transcription hypothesis directly. Is there a way, one might wonder, that you can add some chemical that might work on bringing either the modifications or the co-activators that are not binding correctly and make them do it correctly? It sounds very opaque, but there's evidence that you can do that. And, we might even hear about that later on today. And, if you can do that, then the transcription works better, works the way it should, and you don't get the disease. A third way would be to protein structure control or increased protein degradation. Remember, if the HSPs, these chaperones are binding, it seems like it folds in a way that can be destroyed. What if there's a way of making this one bind to HSP? Maybe we can force it to be destroyed correctly. And again, there's evidence that that does seem to work in mice and cells. Or what if there's another way of getting rid of the androgen receptor? And that's where that autophagy problem. So are there something we can give to the cell that makes it increase the ability to send the androgen receptor to autophagy so you don't get those toxic fragments laying around the cell? Again, there's evidence that this seems to work in cells and mice. You can work by just removing the AR. If you didn't have AR, you don't have any problems. At least no problems with KD. Well, it turns out there's a way to do that. There is something called, I love the name, antisense oligonucleotides. I mean, God, man, that's a cool name. You don't hear it as ASOs. What ASOs do are they're small pieces, small chemicals that bind specifically to a specific RNA. And you can pick which RNA it binds to. So if you pick it to bind to the RNA from the androgen receptor, the cell, it will bind to the RNA and the cell goes nuts and says, I don't want this here, and it'll destroy the RNA before you ever make the protein. So if you add the ASO and it's against the androgen receptor, the levels of androgen receptor drop. This sounds amazing, and it's worked. There is a drug now on the market against SMA, which is a much more severe form of muscular atrophy that works based upon ASOs. Plus, there are mouse models that have been cured with ASOs. So this is a pretty, it's very specific, it's pretty neat. And of course, the last thing we can do, and this is the one everybody who's a patient loves, gene editing with CRISPR. Because this is the only one that cures the issue. If you can gene edit, you can go in and cut the CAG repeats, pull them out, and bring my 46 down to 30. And then I won't have the disease anymore. This is ideal. Well, almost. The problem with CRISPR, two problems. One, it's specific, but not that specific. So, for example, let's suppose, it can go to the androgen receptor 999 times out of 1,000 and go somewhere else 1,000 times. You're going to say 1 in 1,000, that's nothing to worry about. Until you realize that a single arm muscle probably has maybe 1 million copies of the protein that you have to change. And so just in that muscle alone, you're looking at 1,000 times it goes to the wrong place. It makes a change, and that change could be disastrous. When you're talking about doing it to the whole body, it just increases the chance, and it's almost certain that something bad is going to happen because it's just not specific enough. Now, they're getting better. So if you read the paper, they'll say, oh, well, now we're more specific than we used to be, but it's still not perfect. And to do it in the whole body, you almost need for it to be perfect. Second, the machinery is very large, and it's hard to get it into cells. So it's another problem of trying how do you go about doing this. Again, they're working on it. I expect both problems will be solved. They're just not solved yet. And I know there's always, why aren't any of the KDA researchers working on this? And the reason is because they're not, and I may be wrong with some of them, but most of them are not CRISPR experts. They're KD experts. So they can handle KD. There are, trust me, more researchers working on CRISPR to make it right than there are KD researchers. So you don't have to worry about people not studying CRISPR. They're there. And they don't get there. But in the meantime, we have the other possibilities. How much time? 10 minutes. Maybe. I think I could do this in time. First of all, because that's where I'm going to end if I didn't have time. What can you do to help? and you've heard this before and you're going to hear it again, two things, at least with regard to patient groups. One is you can donate money. We always can use money. Good thing. And the second, and maybe to some extent for Kathy, would be donate time. We need people who can help put together things like this conference. And the more people who help do it, the less work each person has to do. So it's really important, I feel, that we try to, if you want to do something, this is a fantastic way to do it. With regard to funding, this is a list as of, I think, last year of all the people who have ever received the KDA grant. Some of these people are in this room. Some of the people, we have two groups from pharmaceutical companies who are going to talk about possible treatments. Both of those, we can find their origination by the people in here. It's, very important. So we do a really good job, I think. The second thing you can do is sign up and complete the Global KD Patient Registry. It's going to be about 40 questions. It's about 40 questions. They're pretty easy, easier than my test would be. And it takes maybe about 20 minutes to fill it out. And, there's a couple of reasons. One, I would recommend that you say that it's okay to share the data. You will never be associated with the data. It will be only in kind of groups of data. So they do respect the privacy. But the reason it's important is that we try to update these every year. And what it does, it gives a natural history of the disease about how you feel about it. So in 5 years, a researcher can look and say, look at how this changed or how this changed in this person. So it's really important, I think, in a very easy and inexpensive way to do basically a research project. Second, it's also a source of names for clinical trials. So I just got my message from Angela about 3 weeks ago about a trial at clinic. Great. Yes, you can, and you should, and that's the whole point. You'd like to hear, so yeah. So let me do one last thing, and hopefully we then have more time for questions. One of the things you have is how do you know what to test? How do we know what works? And I look at it that there are 3 levels of information. The first of these, and there should have been another slide, but we can use this. It's anecdotal reports. And we had a lot of that yesterday when we had the people up here and they tell you, well, I take this, I take this, I take this. And they're very powerful. The problem is that they've never really been tested. And it's tough to know, do they really work? So it isn't a question of not taking or taking them. It's really a question of, if you take them, make sure they're safe. So if you take a supplement, I would try to make sure you talk to your physician, especially if you take any other pharmaceuticals, because some supplements do interfere with other pharmaceuticals. So just want to be sure that you're not hurting yourself. Now, the second one is animal and models, which are known as, I thought I misspelled it. Preclinical trials. So this is where they test on mice, fruit flies, etc. It's really important because it's a way of testing a wide variety of things relatively inexpensively. The problem is mice and flies are not humans. So just because it works in mice doesn't mean it necessarily will work in humans. And that transition is really difficult. I'm pretty sure Kurt, when he talks, is going to say, if we were mice, we'd be cured by now. But the reality is that when we try to transition that into humans, it never works. It has not worked so far as well as it does in mice. So that means we have to go one step further and do clinical trials. And clinical trials are in different phases. You'll have about phase 1, phase 2, phase 3. Phase 1 and phase 2 are somewhat similar. They're both not looking really to see if the drug works. They're working to make sure the drug doesn't kill you. They want to make sure there's no side effects. They will look to see if it works, but usually they're much shorter. So in Kennedy's disease, you really need to go a couple of years. These are maybe 6 months or 6 weeks. It's not enough to really test efficacy. What they're really trying to find out is to fine tune how to give the drug, how much to give the drug, and to make sure you're not going to get harmed by the drug. Once it passes those, we head off into, goodness, I don't know what I did. Anyhow, we go into phase 3, which are the real clinical trials. That's where they get a big group of people, half of which get a placebo, half get the drug. No one knows who has what, and we follow them over 2 years. And usually maybe 50 to 100 individuals. If it passes that and the FDA likes it, it becomes a drug. Then they still follow, because it's possible that there are side effects that are rare enough that aren't picked up in the phase 3 trial. So they do keep following to make sure that nothing else pops up. And and so the very end is just, and this will be the last thing I'll talk about, is I believe this was for Parkinson's disease. I could be wrong. In 15 years, they had 99 clinical trials, 41 compounds and interventions. 25% went from phase 1 to phase 2. That's 1/4, not that many. About 19% got into phase 3, and from 2 to 4, only 14%. Now, I'm not 99, I mean 14% is on the order of like 5. So only about 5 got to be approved. And it's an expensive process. So when the people come and talk about clinical trials, realize it's expensive, and it's really helpful that we volunteer. And I'm going to stop there in case anybody has any questions. I hope you can hear me. I tell you, I worry about my voice these days.
You were great. Thank you, Ed. We always learn a lot from you. And we could listen to you all day. I'm sure I could, for sure. One question.
Let me.
Kathy wouldn't let you. That's right. It's your fault. One question came in from online, which is a common question I think we've heard before, and many people will benefit from your response. And the question is, if over 35 CAG repeats means you have KD, I have 45 CAG. What is the numbers for the high end of the spectrum? Does the low end of the spectrum mean your KD is not as severe as the high end?
And someone who's not an expert, the high end is really usually in the 50s. I think the highest is 60, 64, but there's only been like 1 or 2 of them. I would say the average is in the high 40s. With regard to severity and onset, there's a little bit of a correlation between the more repeats and the sooner you get it, or the severity, but it's not predictive, because there's so many other factors apparently that if I told you I had 46 and someone else had 46, we could have completely different symptoms. So it really doesn't have as much meaning, the way I see it, as it might be in Huntington's, where I think there's a stronger correlation. And Kurt, would that be okay? Yeah. And so that, yeah.
I think it just looks better in Huntington's disease because there are a lot more patients.
Yeah, and that too.
But there's a lot of variability, so you can't predict exactly when it's going to come on or how severe it's going to be based on.
My interpretation is that there are a lot of other factors in the cell that make it variable, and we don't know what those are. I mean, why is it I'm at 68 and I can still walk really well, and other people at 35 are in a wheelchair? I mean, it's a very weird disease.
There's also just random chance,
Yeah.
Good luck, bad luck.
Is there another question over there?
More out of curiosity, because I don't know what my CAGs are, because when I tested way back when, I didn't think, I was just told I have it. Okay, if there's a carrier that has, let's say, 2 boys, and one test has 40 repeats and one has 32 repeats, is that what would tell somebody that they don't have it?
Yeah, and so realize that women have 2 copies of the gene because they have 2 X's. So, and they won't have the same number of repeats in their 2 genes. So they got 1 from the mother and 1 from the father. So they could have 30 repeats and 45. And then there's a 50-50 chance of which one they give to their sons. Okay, so that half the sons on average would get the 45 repeats and half would get the 30 repeats. And I mean, it's statistical, you can't predict. But
Like I said, my brother has it. He tested a while back, and I don't know what his repeats are, but we're just two brothers that were the only siblings.
right,
So we can
it's
both,
just
it's random. We can both
it's
have
random.
40 or
So I have a brother, and he did not get it. So, and it's like getting eye color if you have, you know, anything like that. It follows regular genetics that you learned in high school. And it just happens. I mean, and it could have been, you had just as good a possibility of most of you not having it.
Okay, I think we're going to have to move on in the interest of time. For those of you who have questions who are in the conference, Ed will be around for the next day and a half. For those of you online who have questions, go ahead and submit them in the Q&A and I'll make sure Ed gets them. Okay, we're going to move on to Beth Solomon. Beth, are you out there? Are you online?
I am online. Can you see my screen?
We're good here. So let me, while that's loading up, introduce Beth Solomon. Beth is a lead senior speech language pathologist at the speech language pathology section of the Rehabilitation Medicine Department at the NIH Clinical Center. She received rave reviews for her talk on swallowing last year and the year before on speech and swallowing. And she gave us a wealth of information in those presentations. Today, she's going to focus on practical exercises you can do to help in both speech and swallowing. So I think we're ready, Beth, when you are.
I hope you can hear me. We are going to be talking this morning about speech and swallowing ideas. And with me saying that, the views that I'm going to be talking about and the things that I'm going to be saying are actually my views. They are not views expressed and reflect anything official from the United States Department of Health and Human Services or the National Institutes of Health. So if I'm talking about other trademarks, logos, anything like that, it is not constituted or endorsed by the federal government. Any exercises, strategies, positions, maneuvers that I am going to talk about or demonstrate in short videos, again, are ideas that I have come up with over the years to help facilitate improvement with both speech and swallowing function. And I urge each and every one of you, if you have difficulties, to seek a professional. So as I move on, we are talking about speech. We are not going to be talking about hearing. However, it's important to know that people need to have good hearing to ascertain whether you are intelligible or not intelligible. So that constitutes family members making sure they have their hearing tested, especially husband and wives. Over the years, the husband tunes off or the wife tunes off. So make sure you test your hearing as it relates to listening to one's speech. We're going to be talking a little bit about swallowing and how we may improve. Now, both functions, speech and swallowing, are very mundane type functions. It's something we do every day. And when something arises and it's not what we think is normal, it can be very discouraging and frustrating to the point that people don't even want to venture out or eat with other people or speak with other people. So as I said before, I encourage each and every one of you to seek the professional for any advice. See your speech pathologist, neurologist, ear, nose, and throat physician. I'm also going to state right from the start that I do not have one recipe that will solve everybody's problem. I've been practicing as a speech language pathologist for over 30 years and see a variety of different types of disorders at NIH and in the previous places that I have been. And I have never, I believe, prescribed the same thing in the exact same way on 2 occasions. We're all different, and that's what makes my life and my job very, very interesting as both a clinician and a researcher. So some general tips as it relates to your speech. keep with a team. Find your team of people, your professionals that you can go to and get some advice. Yes, Kennedy's is a rare disorder, and sometimes your speech language pathologist may have never heard of it, but they can check out and see where they can find more information. It can be a puzzle in the beginning, but sometimes thinking out of the box and working with others who are knowledgeable can be very helpful. And once again, for both you and the therapist, sometimes it's a balancing act. We all have our lives to live, families to raise, and having to do therapy and exercise is sometimes difficult, and we have to pick and choose which is most important to us. So as we said earlier, there's no cookbook recipe. Exercise is good, but I would avoid exertion and overwork. Again, seek your consultants, and don't just seek them one time. If your status changes or you think something's different, go back and get another therapy program. It's very important because it is a neuroprogressive disease and it does change. So we have to have modifications. So if we start off just with speech production, how is speech produced? We obviously are inhaling through our nose and our mouth. The air goes down through the back of our throats, through the larynx, which houses our vocal cords, to our lungs, which then inflate even further. Our diaphragm collapses and actually moves down, allowing more air in. It is then the air coming back up that powers these two vocal cords, and you can see the tracheal rings here. These are the vocal cords at rest. They are then vibrating, and for men, that can be anywhere from 80 to 100 times a second, and that produces the sound source. That sound source is considered the engine, or that's what I would call it. The actual gasoline to your engine is your respiratory support, or your breathing and your exhaling. The air then travels further up through your nasal passages, and this is where we get the resonation or the nasality that we talk about. Again, it's your lips, your tongue, your teeth, this air, the engine that actually produces the speech. A B sound in the front, a T in the middle, a K in the back to make certain words and for those words to be intelligible. All of these things have to work together. If there is disruption in any 1, 2, or 3 areas, the speech is going to sound impaired. It may be a combination of areas. You may have only one area that's a problem. But when addressing your therapies, we have to think about all the different systems. Now, I'm not going to be talking about the central nervous system in terms of the language function and word retrieval. That's for another talk, another day, but I will be addressing therapy as it relates to the respiratory system, the laryngeal system, the resonatory, and the articulatory. The easiest way for me to break this down is by system, and then utilizing a combination of exercises to make your recipe and everybody's recipe be individualistic. Again, I'm not talking about the auditory system. That needs to be checked both for yourself and your family members or the other people that you are communicating with. So again, we have the respiratory system. It is impaired, and how in Kennedy's we find that that usually shows as reduced expiratory force for phonation or the voicing that we just spoke about. This results in short phrases, diminished volumes, and a real feeling of work, especially when we're tired. A lot of this is related to the weakness and phrenic nerve weakness and less air coming up and through. So if I was to have my toolbox and I was just to address the respiratory system, I'm actually looking at breathing exercises. I may be looking, and I'll be talking about this in a minute, inspiratory and expiratory trainers, and then basic traditional coordination of your exercises in breathing and applying voice to that. And once you apply the voice to the exhaled breath, you'll apply speech. And we also can look at our physical therapy partners. Joe will be speaking next from NIH, and they can also help us with some core strengthening. So in terms of breathing exercises, I have 2 diagrams right here, and I look at abdominal diaphragmatic breathing to begin with. This can be done either sitting or are laying down. I prefer starting a patient laying down, where the patient actually places his hand on his tummy and on his upper thoracic area. And the first thing that I would have any patient do is actually push down on the tummy and exhale at the same time, just to get yourself in a rhythm for inspiring. What you would then do is then inspire through your nose and your mouth. And as you inspire, your tummy needs to raise. As your tummy raises, you have actually brought in the air into your lungs, and then exhale slowly again, pushing down on your tummy and exhaling that back out. I would repeat this in a series of 10 exercises really slow, and do this multiple times a day if you can, just for short periods. And when I say short periods, I'm talking under 10 minutes of time. And just be careful if you are laying down, that when you do get up, that you sit for a few minutes, because sometimes when you're doing some abdominal breathing, you get a little dizzy because you're really exchanging of a lot of oxygen back and forth. But it's a good respiratory practice for you to do. You can do it like when you wake up in the morning, when you go to bed at night, or if you're taking a rest on the couch later in the day. I mentioned earlier, inspiratory trainers. Many of us may have seen devices such as these on the left. And when you're in a hospital and after a surgery, and they ask you to blow and raise the bubbles or raise the meter. What we're trying to do for inspiratory training is to really expand those lungs, bring in the air through the nose, or I'm sorry, through the mouth, and then expiratory them back out. Now, the difference with an expiratory trainer, it does have a passive resistance that you can actually change. And I use them quite frequently as part of my therapeutic regimes with patients. You can see here a man who does have some difficulties actually closing his lips around the mouthpiece. So he does use his hand, and he would be blowing out or during an exhalation cycle, and it actually makes a little bit of a noise as you're doing this with the resistance. And as you increase the resistance, we then get a little bit it gets a little bit harder for the patient. So what we do is we do this over a period of time in combination with modulation, as I said earlier, of our breath with speech. So I may start with using the expiratory trainer. At the same time, after we've used it for a few cycles, we would then maybe use a couple of key phrases or a couple of key words that they would use, and making sure they can apply the word onto the expired breath. We find that many patients sometimes will talk on what we call residual air and not really place the words completely on the exhaled cycle. So that's really an important part of all of this in terms of therapy, and your speech pathologist at home can help guide you through some of this. I'm now moving on to the laryngeal system, and here are the vocal cords at the top, nice and open, and then you're seeing them closed. But on the bottom, this is how they would be vibrating. It is a very low volume. And with Kennedy's patients, what we find is that voicing is often demonstrated as decreased volume or a gravel quality to your voice. You may have some hoarseness, or you may have a strained vocal quality. Sometimes there's even some wetness or gurgly to a voice, and occasionally even a tremor. A lot of this is related to the vocal fold atrophy. The vocal cords are actually a muscle, the vocalis muscle, and it's surrounded in the larynx with other muscles and cartilages. And so there may be a small gap when you go to phonate or actually voice, and that gives you the breathy quality, and that is perceived as decreased volume. Or you may lose a lot of the air from diminished respiratory support or in coordination, and then again, you don't have the volume or the strong engine. So if we are looking at our tools in terms of voicing, the first thing I would always do is check with your ENT, depending on the type of voice production problem you have, just to ensure that there's no growths or lesions. I would start you on a vocal hygiene program. Yes, it's similar to dental hygiene, but it's for the voice, and I'll talk about that in a minute. And again, going back to the respiratory system, we coordinate voice with breathing, and then we add speech on top of that with easy onset and easy flow, and then modulate that in a systematic fashion to conversation. If needed, we would also use amplifications. So if I go one step further and just say, what is everyday voice hygiene? That means stay hydrated. Hydrated doesn't mean coffee and tea and Mountain Dew and drinks such as that, because those are dehydrating agents. We want water, sometimes water with lemon juice, because that often cuts the viscosity of our secretions. We would have you reduce your high intensity voice, such as yelling at kids or at work or other types of, unfortunately, the rage that happens in this country. We would have you also avoid any types of allergy and allergens, even antihistamines, they're drying agents. I would have you manage, I can't see all my slides, hold on a second. Sorry, and manage your reflux. Many patients, as we get older and with Kennedy's disease, do demonstrate some gastroesophageal reflux. So making sure that you're on an appropriate protocol for reducing that, because we don't want secretions coming back up through your esophagus. And we'll talk about that a little bit later when I get to swallowing. So if your speech and your voice sounds like it has a kinked tube, again, you're going to work with your therapist for coordinating that exhalation and speech. So you may start with easy vowel onsets or easy words and expanding that to short phrases, and then modulate that possibly with the reading or with day-to-day conversation. As it relates to intensity, if I've already alluded to making sure your players all have good hearing and don't need any kind of amplification on their end from their hearing standpoint, I would encourage you all to utilize positioning to the best of your ability. Positioning, whether that be at the dining room table, and making sure that you are seated in a good proximity to the ones that you're going to communicate with. You don't want to be at the end of the table and having to shout to the other end of the table, especially as we're hitting the holiday season, or if this is in a work environment as well. Position yourself in your living rooms and dining rooms and turn off the excess audio visuals, such as your televisions or the computer games, because you have to talk over them, and that's sometimes very difficult. Often I will use a decibel meter, and that's a way to actually assess in numeric form the decibels of your voice and see where you're at, get an idea of what is comfortable, and you can monitor that over time. And I use that also in therapy with my patients to look at the volume of their voice, because you can get an idea perceptually of what is comfortable and how much effort it takes to be comfortable. And we use that actually in Parkinson's disease. And it actually is a quite useful technique over time to work with patients where they can actually have that visual feedback of what is the actual number. If all else fails, there's all types of amplifiers that you can wear. They are very small these days. They used to be a lot bigger. And when it comes to telephone aids and getting heard over the telephone, there's all types of devices that can amplify your voice. It may not improve the clarity or intelligibility of your articulation, but if your voice is softer, this is a way to amplify it. When we talk about resonation or the nasality system, we're actually talking about how the acoustic properties, which are very complex, the air moves up through the hypopharynx, and the soft palate or uvula, with what you're seeing here on the left, would actually close. This is a two-dimensional model, and we know that some patients with Kennedy's have pharyngeal weakness, neck weakness, and again, decreases in the respiratory support. So what happens is, along with some of those other characteristics that I've previously described, you may then have more nasality in your speech. And I believe you can see this is now the actual view of your soft palate. This here, you can see it's a valve. In the front, what you're seeing is the uvula closed, and on the sides of the pharyngeal walls for the complete closure. It just gives you an idea that if you had a leak in the hypopharynx, you would have little air coming through an opening, or there may be even some fluttering of some mucus. So when it comes to doing exercises for strengthening or at least maintaining the strength of the hypopharynx, we can utilize production of back consonant sounds in combination at the word level and then at the phrase level. We can utilize different diameters of straws, and I've shown you those here, where you are actually pulling up and trying to strengthen, and the lower the diameter, meaning the smaller the straw here, versus the bigger straws, is much more difficult to pull up, which will cause some actual strengthening if we can get you doing this on a routine basis. And if it is that severe, we would possibly move on to dental prosthetics. And these are devices that can be made by a maxillofacial prosthodontist, and they can be very beneficial for both speech and swallowing. The one concern that I have is it does require flexibility. There are some insurance constraints, and you need to talk to your individual provider up front. But what you're seeing here is an actual prosthetic device. It's on a retainer that gets hooked onto your teeth here, and the prosthetic device actually pushes up onto your palate, so there's less leakage coming out. Our last system as it relates to speech is the actual articulation. This is the part that people complain about the most. They're not clear, they're not as intelligible, the sounds are distorted. Some people say I'm slurred, I sound like I'm drunk, sometimes I'm lisping. Well, what happens here is that you need to have this coordinated system that I've described before. And a lot of times with KD, there's cranial nerve weakness in terms of moving your articulators. That would be your lips, your tongue, in a very fast pattern. That's secondary to the muscle atrophy. And then there's discoordination between the systems to make those sounds, so the speech and the articulation actually comes out impaired. So if we're looking in this system, some of the types of things that I would work on is trying to maintain the strength of your lip, your tongue, your base of your tongue, your palate. That can be done with exercises. And again, a small amount of exercises. We're not talking about overexertion. They are tongue strengthening devices, the Iowa oral pressure instrument, and I use that for measurements for patients over time, and actually can see how strong someone's tongue actually is. I do recommend gum chewing. We'll see a short video a little bit later. We'll do some specific sound therapy and speech therapy with sounds in words and phrases. I use a lot of reading therapy, conversation, tongue twisters, and I really try to think out of the box. We may also use other feedback loops. This is a picture of the IOP that I spoke about earlier. This is a tongue bulb here that's air-filled, And what happens is you actually put this on your tongue and you can actually measure and get a kilopascal measurement here. And this can also be utilized, and people have used this in different other diseases for increasing tongue strength for disorders of swallowing and speech. This is a video, and I apologize. It is of one of my research students last year, and I'm apologizing in advance because she is a woman and not a man, and I would have rather had a man, but I had a woman student last year. We are going to be doing some lip exercises, and what I've asked her to do is pucker as hard as she can and getting a complete lip closure. You see there's no holes in the lips, and I'm asking her to hold it for 10 seconds, and she's counting 1 Mississippi, 2 Mississippi, 3 Mississippi, And she went all the way to 10 while I was talking. And we do that repeatedly, usually 10 times. In the course of one trial, and I usually will have you trialing this at least 3 or 4 times a day. That's one basic exercise. The second is more of a, oh, I'm sorry, let me just stop her for a second. Let me bring up the root beer candy. I've actually spoken with some of my KD patients recently. I came across the root beer barrel candy exercise where you're actually holding the root beer barrel in between your lips and you're puckering with it. It actually gives you a feeling of something there. You can hold it. It's a little bit harder. And after 10 seconds, put it in your mouth, savor the taste, swallow the taste, and then repeat it again. This is another exercise that works well. And as the root beer candy starts to melt down over time, you're going to find that it is a lot harder to do. So it's just a feedback with the actual exercise. And I have a lot of KD patients doing this as well. The lip retraction exercise is exactly what it says. It is pulling back your lips as hard as you can, holding for 10 seconds like before, 1 Mississippi, 2 Mississippi, 3 Mississippi, and then relaxing after that. What's important with this is that we use a visual feedback such as a mirror, because you can actually see when you start to weaken over time, and then pull back if you need to. If you don't use the feedback, sometimes you miss where your weakness may be. Tongue protrusion is the next exercise in terms of moving your tongue, and people don't realize how difficult it is for you to actually stick your tongue out, hold it for, again, that 10 seconds, hold it way out, don't let it move, and you'll see Andrea's going to do that. She's got her tongue out, her lips are open. You don't do it closed because the lips will help if you do it closed. You really want to isolate it, make an isometric move. And you'll even see Andrea, you see her tongue is actually moving and fasciculating a little bit up and down. She had a hard time doing this and didn't realize that she has a little bit of tongue weakness. But it's important for you to try to do this again a few times a day in series of 10s is always what I recommend for people. I'm going to move quickly on to swallowing. Swallowing is a very important function, and anybody who has a swallowing impairment knows that it can be very disruptive and frustrating. We actually are swallowing typically 600 times a day, more when we eat, less when we sleep. The New England Journal of Medicine actually in 2001 stated that aspiration pneumonia is the leading cause of death with people who have swallowing problems and other neurological diseases. So that was a very important finding way back when. And we know that about 80% of the Kennedy's patients exhibit some kind of swallowing problem or dysphagia during some portion of the disease process. Swallowing, as I'm going to describe it to you, is in 3 phases. It's the oral phase, the pharyngeal and laryngeal phase, and the esophageal phase. You're seeing above just one repeated clip of one swallow where you see it moves very quickly through the mouth at less than 1 second and through the pharynx at 1 second down into the esophagus. It's very well controlled. And again, if we're weak or there's discoordination in the timing, things can go the wrong way and we would end up with laryngeal penetration or aspiration. So as a speech pathologist, we manipulate we watch you manipulate food, forming a bolus here in your mouth, and actually a series of reflexes will kick off so no food comes out of your nose. We watch how the constriction goes and movement through the hypopharynx, the back of your throat, and making sure nothing goes down the wrong pipe, and that the upper esophageal sphincter actually opens and food gets traveled down. One thing to always remember is once the upper esophageal sphincter opens, because it is a closed sphincter, there's a series of peristaltic movements, that's kind of timed movements that move the food down into your stomach. These time movements, if you have reflux, can come back up and it can set things into disarray, and acid and other gastric contents can come back up and move over into your vocal cords, setting into spasms for patients. And we've talked about this previously. Patients have some laryngospasm. I spoke of laryngeal penetration. That is when any food, substance, liquid, or even some of your secretions can fall into your airway. That's what you're seeing here. And then there's full aspiration, what you're seeing down here, where it's gone past the level of the true vocal cords. What's interesting over time is when you have laryngeal penetration, you are actually clearing your throat a little bit more frequently, something like that, or coughing. When you're fully aspirating, you're coughing more. If you have silent aspiration, which is what I referred to earlier, that's when there is no sensation. And typically we're seeing that more and more diseases when patients have started to aspirate, kind of gone for a period of time, they didn't realize they were aspirated, and then their body becomes desensate to the material that they're aspirating. So it's more of a concern, and we usually pick it up on a swallowing study. So general tips when we're talking about swallowing: if you're tired, when it's eating time, go for softer, easier meals. Easier meals does not mean salad. Salad is one of the hardest things to chew and get through and swallow. Avoid larger meals if you're fatigued. Consider eating earlier, such as your bigger meal in the middle of the afternoon, not later in the afternoon. Please avoid things like rice, popcorn, dry ground beef. I said lettuce, raw vegetables, they're very difficult to shear through and move around, especially if you're tired. Mixed textures such as grapes, honeydew melon, watermelon, where you actually bite and there's a squirt, can sometimes cause difficulties, as well as vinegar-based products and salad dressings. Mixed chocolates, chocolate-covered cherries, as we get into the Christmas season, often have caused people to have great difficulties. Basic swallowing rules: upright seating during meals, very, very important. Avoid distractions. Those are your TV games, just difficulties. I would shut all those things off when you're eating, because any distraction can really cause a major problem. For patients that have had some trouble with liquids, often a clinician and a speech language pathologist or neurologist will tell you to tilt your head forward or do the chin tuck method to help protect your airway. Just make sure you take a sip, chin tuck your head down, swallow, give yourself a second before you raise your head right back up again, because anything that may get stuck in the vallecular space, which is between the tongue and the airway, I don't want it to fall right back into your airway. So give yourself a little bit of time and stay upright at least 30 minutes following your meals. Don't just go lay down and watch TV or the sports or the news. You got to stay upright after you eat to avoid some of this reflux that I've mentioned. Andrea is back with us. Some exercises for, depending on the problem, especially if you're having trouble manipulating the food, is some tongue lateralizations. And what you're seeing on the right-hand side of the screen is some Cheerios or chocolate-covered morsels that we have in chocolate chip cookies, where Andrea is now placing the actual chocolate chip on her tongue, holding it again out for 10 seconds. We're utilizing the cereal or the actual weight, the cereal or the chocolate chip as a small weight on your tongue. And she actually, as you're seeing here, has trouble even with this. This is another exercise. You're holding it for 10 seconds and repeating. And I would have you talk to your speech pathologist about something like this as well. Another exercise, as you see here, is gum chewing, and I talked to lots of patients about it. It's a nice exercise because you're actually chewing the gum. All right, let me just talk a bit. So the gum chewing is really, she puts a piece of gum in your mouth. The top one is like a Trident type of sugarless gum, or you could go heavy duty or work your way up to a bubble gum. You put it in your mouth. You chew it from one side to the other side. You're swallowing the saliva that does accumulate. It's a nice coordinated task. You're only chewing for 10 minutes. So I don't want you thinking that, oh my God, my therapist told me I have to chew gum and I'm chewing gum like all day long. No, it's a small task, no more than 10 minutes. one piece at a time. As you are able to chew one piece for 10 minutes without any kind of discomfort or any kinds of problems, I will add a second piece. I will add a third piece. It gets more and more difficult, and especially as you move up into the bubblegum arena. Another exercise that is used quite a bit for strengthening or maintaining the strength of the base of the tongue. The base of the tongue is very important. It kind of helps propel food through your pharynx, is the Masako maneuver. What you see here is a gentleman. He has his tongue out. You can actually stick out your tongue. You hold it with your fingers or with a piece of gauze, and you swallow very hard. And that would be another way for strengthening. And I would do this repeatedly with a patient who is having trouble with food, feeling like it's getting stuck in their throat. And obviously they've been evaluated by a therapist. For some people that know that their food is getting stuck, it's been confirmed on swallowing study. An effortful swallow is often very, very helpful. An effortful swallow actually looks at a swallow where you put the food in your mouth, you close your lips really tight. She has the applesauce, as you see here, and you pretend as you're swallowing that you're swallowing a grape and you're bearing down very hard, almost like you're having a bowel movement as you swallow, because that helps to increase some of the intraesophageal pressures in moving the bolus material and the food down through the esophagus. It's just another exercise and technique that we use. The last technique that is what we call the supraglottic swallow. I'm going to stop that right there. It's actually, because you won't hear me, the supraglottic swallow actually is taking a bite of food, holding it in your mouth, and as you're holding it in your mouth, what you're doing is you are holding your breath, then you're swallowing, and after you've completely swallowed, you release your breath, you cough, or you can say ah, and you breathe normally again. What this does is it forces your vocal cords to stay closed during the swallow for a couple of seconds further, because we normally, when we do swallow, our vocal cords close, but we want them to remain closed so we don't have anything going down into our airway, such as a small piece of liquid. So that's another technique that I would use. I'm going to go, the last slide here is laryngospasm, which is really a sudden spasm of your vocal cords. It lasts usually less than 60 minutes. I'm sorry, 60 seconds. I didn't mean minutes. It's less than 1 minute. It's very, very frightening. It's when you really can't breathe. You feel like you're choking, you're coughing. Your vocal cords actually do close, and the possibility of GE reflux, anxiety, allergies, vocal fold dysfunction have all been linked to having this. People sometimes will wake up in the middle of the night with a spasm when they're sleeping. Sometimes you will hear this whistle sound. That's okay, because then you know there's a little bit of air coming through. The first thing you want to do when it comes to therapy is ensure that you're really having the laryngospasm, and make sure you talk about this with your physicians right up front. If you can, sit in a chair, purse your lips. I mean, and when I say if you can, because it comes on very suddenly, you're in a spasm, you don't know what to do, you feel like you can't breathe, and try taking short breaths in and out. I will tell people to carry a small little straw with them and put the straw in your mouth if you can, and breathe in and out. You actually can hold, I believe, I can't see it if the last picture came up, nope. You can actually do a maneuver where you're reaching for the fleshy part behind your ear. It's called Larson's Notch, and pull your jaw forward as you're having the spasm. That often will release the spasm. But be sure that the spasm typically only lasts for about a minute, 60 seconds. It will release, and the worst that usually that would happen is obviously you would pass out. But those are some issues that can help, or techniques when you are having the laryngospasm. I don't know how we're doing on time, but I'm going to stop here. I can take questions, or we can talk later during the panel.
Yeah, thank you, Beth. That's a wonderful presentation. As always, a lot of good practical advice. There are a couple questions online, and we have some maybe in the room here, but I think we'll wait until we do the clinical clinicians panel where you will be presenting as well. You'll be available.
Can you hear me?
Yeah, we can hear you. There you are, Joe. Go ahead. So let me introduce Joe. Joe is a senior staff therapist at the physical therapy section in the rehab medicine department at the National Institutes of Health Clinical Center. And you may have heard his presentations at earlier conferences where, like Beth Solomon, he also received rave reviews. And you may have seen Joe's videos on exercises on the website that I just mentioned. Today, Joe's going to provide some updates on exercise in your KD. So Joe, welcome. Over to you.
All right, thanks. We're so happy to be here. I want to introduce myself. I work at the NIH. I'm out in the East Coast. Have probably met many of you, and very grateful for that opportunity to speak to you today. I have Ali Nemec with me, who is a student working with me for 2 years, and we're both working on the KD natural history trial and getting lots of good experience there. We're just gonna kind of go right through this presentation, which I think will not last more than 20 minutes. And we'll hold questions until the end if there is time. The disclaimer is the same as it's been before. I think I'll let you guys read this, but just to quickly state, what we're presenting to you today is not the views of the government or the National Institutes of Health or the Department of Health and Human Services. And probably a little bit more importantly, it's not a prescription of any kind for any single person. It's just presented for educational purposes. Ali's going to start the beginning of the presentation, and then we'll switch back and forth.
All right. Hi, everyone. Thanks for having us today. So today we're going to take you through the process of creating, starting, and sticking to an exercise program. So we'll start with physical activity guidelines, then on to why move. Then
excuse me, guys, are you sharing your screen?
We are.
Okay, because we're not seeing your presentation right now. We're only seeing your camera. There we go. Now we see it. Thank you.
All right.
Okay.
So like I said, physical activity guidelines, why move before starting, how to start, exercise examples, adjusting and monitoring, and then we'll wrap up. So as a starting point, the US government has pulled together years of research to create these physical activity guidelines, which might vary slightly by country, but we believe that these can apply to everyone. So to start, all adults should get 150 minutes per week of moderate aerobic activity or 75 minutes per week of vigorous aerobic activity. On top of this, you should spend 2 days a week doing muscle strengthening. For older adults, there are 2 more considerations. The first is to incorporate balance and functional training. And the second is just taking stock of any physical limitations your body might have as you age. Finally, for adults with disabilities, everything I've said before still applies, but we want to add meeting with your healthcare provider as they can help you navigate physical activity and exercise, and they can help you adapt exercises to best fit your individual abilities. And of course, for everyone, we want to avoid inactivity. So we have all these guidelines and recommendations because it's just so important that we keep our bodies moving. So this slide is just some examples of things you can improve when you're physically active. And we think part of the fun of getting started with exercise is looking at a list like this and picking out things that are important to you personally, and you can choose exercises that target those things.
So before we get into just breaking down the recommendations for the program, we want to just review quickly the limited research in exercise in SBMA in 2000, and it's sort of nice to see all on one page here because it's kind of as it spreads to the right. We noticed that the exercise results have been improving. In 2009, there was a 12-week moderate intensity cycling program that the authors basically concluded that it was not helpful. Moderate exercise did not increase VO2 max. Then in 2015, at our site, we did a functional exercise program. There was still an overall hesitancy to report great improvements, but when you divide it into those who began the exercise with a little bit lower level of function, we did see a significant improvement in function compared to the control group. So we felt pretty positive there. Then in 2019, you really get to a much nicer result type study, albeit a small one, a cycling program that is looking at high intensity interval training. And there was an improved VO2 max. And importantly, the participants chose to continue with that exercise following the 8-week high intensity training program. And then we have a couple of case studies, which we know are much lower evidence, but it's just nice to see these studies coming up as we recent studies that are coming up. We'd love to see more of that. Before starting, some considerations are, we really advise you to get your current overall health status. Keep it in mind and have any, if you have a cardiologist, if you have cardiovascular disease or arthritis, you need to kind of really take stock and get these evaluated to make sure there are no contraindications to the particular type of exercise that you that you're considering. Then we suggest even before getting going, you might want to go see a physical therapist or a physiatrist or a personal trainer, etc. To think about the things that have been barriers for you in the past, so that you already kind of have a real good idea of what's worked for you, what hasn't worked for you, what are your big concerns that you know about. Do you have drop foot, for instance, or do you have really weak quad muscles, etc. And then individual physical abilities, just trying to really kind of be specific about what things are happening for you. Also will help you to have a framework when you're working with that professional to set up what's going to be most successful. We would also highlight that on many of these slides, we've kind of put a little note at the bottom. So this one here, like for instance, to get to know your own muscle weakness around your foot and ankle, there's a nice foot and ankle video that kind of discusses that in detail on the website. Discuss all of these with your PT or physician, and like we said, we're just trying to recommend kind of getting your ducks in a row. We put a little test question in here. Which of these muscles tends to be weak in individuals with KD and responsible for preventing ankle sprains? We're really just trying to highlight here that all of these muscles have different features and functions. And so your tibialis anterior on the front of your is a common muscle, and it prevents foot drop. And if it's weak, it's important to know that because it can cause tripping and falls. Your ankle plantar flexors contribute to balance and walking, push off during walking. We're finding more and more research that's pointing to these muscles as really being helpful for balance. Quadriceps are the large muscles on the front of your thigh that prevent your knee from buckling. Hip extensors contribute to basically all upright movements, including sit to stand, step up, etc. And the answer to the question is the ankle everter group, which is on the side and prevents ankle sprains. These are the kinds of things that we hope when you go to see a professional, you will get these examination procedures. And again, some of these are in the videos that we've provided in the past. So we said, now we're getting to how to start. We have a little acronym, SAFE. SAFE stands for safety considerations, which may include, getting to know whether you have a high falls risk, what your fatigue is like. Does it come on quickly without any warning? And making sure that you don't overexert your muscles early on. Checking your ability. This is kind of a tricky one. We're going to show some examples of exercise here in a few minutes. And we've kind of broken them down into those with sort of high, medium, and lower levels of function. But it's not always intuitive to know kind of where you are. We have a tool called the AMOT, but again, I feel like visiting a physical therapist and sort of getting an evaluation may help you to kind of zero in on what your level currently is. And then the exercise, if you want to stick to it, we obviously want to make sure it's not a chore and it's not that it's fun. And then finally, what's your end goal? This is something that you may already know exactly what you want to do, but we're always surprised a little bit when our patients come in and we ask them, like, what do you really hope to get out of this? And they don't always have a clear picture of it. So again, sometimes discussing that through, you can really come up with some realistic goals that are very attainable in a short amount of time. And again, you'll note there's a little video on balance and how to start that. Going forward. Now we have a couple of precautions we want to talk.
All right, so this is a slide we really want you to sit up in your chair for and pay attention to, because we do have a couple exercise precautions, and the main one is, of course, we want you to exercise, but we don't want you to overexert your muscles. This can happen in two ways, either overloading, so you're using too much weight, or overusing, which means you're using the muscle for too long of a time period. And this is really important for you all to know, because if any random person were to walk into a clinic or a gym and ask for help starting an exercise program, that trainer is automatically going to start overloading your muscles and start working you to exhaustion. And that's because in typical muscle, that's kind of a way to start causing little micro tears in the muscle. And these little micro tears are damage to the muscle, which causes repair proteins to come in, and the repair proteins are able to build back the muscle bigger and stronger. But we don't know if KD muscle can repair this damage the same way typical muscle can. So that's why we have this precaution, is we want you to be aware that this is happening, and we want you to be able to go to a new trainer and say, I do want to exercise, but I need to be cautious and not overwork my muscles.
So recommendations for getting started are, no matter where you are in your exercise, whether you're new to exercise or whether you're a veteran, whatever you're starting though, you might even be just starting after a time off period. Be conservative initially in the exercise dosing, the amount you do, the intensity that you do, et cetera. You don't have to just go with that first, slide that we showed that shows the whole recommendation for the frequency and so forth. So we want these next three recommendations. We want to look at pretty much everything on the left side and sort of stay away from training to maximum or to overexertion. We really recommend that when you perform these exercises, you do so in a controlled manner. There's a core video and a 10 RM video that, if you haven't seen, each kind of lead to both of these. The conservative initial start, you can be sure if you use the 10 RM feature, which just means whatever exercise you choose, you can do 10 repetitions of them without fatiguing or your muscles failing. And the slow controlled exercises, we talk a lot about using your core to help to provide that slow controlled movement when you're doing any exercise. And then finally, a lot of people think exercise, when you first think about it, the universal thing is to grab a weight in the arm and work the bicep. While there's nothing wrong with that, we really feel like it's best to do multi-joint functional tasks where you can work all of the muscles that were in that so-called cheat sheet that we showed earlier that showed all 5 of those lower extremity muscles. There are several different functional tasks that work them all at the same time. And so you're working on coordination, you're getting a little bit more benefit, not just strengthening, but also improved use of the task. Some examples of body weight exercises, we had, our study had a lot of this type of work in it. So did both of the case studies that we presented that are currently in the literature. Just want to just mention that, sit to stand, stairs. These are all things that we want to do. We want to roll and get up from the bed. We want to be able to turn, step up, stand, use a curb. But you might also just gravitate towards something along the lines of light Tai Chi or yoga. But the whole point here is that you're trying to control your body and moving it through space in meaningful ways to you, and you're doing so in a safe manner. So some examples, we just in the length of time that we have, we just don't have any time to explain each one of these exercises, but they're all vital and valuable. But we're going to mainly just sort of zero in on the exercises for the lower functional level patient or someone who's just getting started. And really what I want you to take home from this is just that you'll notice in all these slides, there's increased support. So gravity is either eliminated from the activity. There's a person supporting a limb as the person moves, or there's a tabletop supporting the limb or some other device or use of a strap, et cetera. So there's a lot of ways to exercise with this extra support, using an assistive device to do walking, or using an AFO, or other examples of how to use some support to continue to get these exercises successfully completed. As you move up into sort of a little bit more middle functional level, we start to add more like resistance training, some light weights. There's a little bit more on foot activity. You're starting to really work on balance. Not that these are all exclusive, they are not. You can be working on lots of different things, no matter what your level is. And there's a little calf video that shows how to stretch and strengthen your calf muscles on the site. And then finally, higher level function exercises. This really opens up a whole list of options. These happen to be a lot of core strengthening in the middle and some isotonic free weights. But as we've shown just recently, it's not for everyone. There's a lot of monitoring that needs to, supervision and monitoring that needs to happen. But weightlifting is an option. We learned that from our patients who were coming into our trials and saying, well, I'm doing it and it's working for me. So we heard that enough. And then we finally got a case study to look at that a little bit closer. One more point, whether you're, whether you believe you're in the lower or the higher or the middle group, again, this isn't to create divisions, but breathing exercises and posture exercises are going to be across the board. There's just no going wrong with strengthening your core, working on ways to make sure that your breathing is as optimized as possible. And then whenever, as you think about, again, we can't tell anybody what to insert into their program. But one thing that you need to think about when you start to insert things into your program is that every exercise, right from day one, you should think about, how do I adjust it up or adjust it down? And there's just a lot of nuance in this. We could talk on this for a long time, but the picture on the left, the example of sit to stand, you can see that this gentleman has a similar chair with a raised height. And so doing the same exercise from the chair to his right would be a little bit more easy, and you'd be able to maybe do those 10 reps. Doing it from the left side is a little bit harder, but the point is, you want to find out what your capabilities are so that you can make those nice movements and repeat that movement 10 times without losing your form. That's just an example of how to move up or down. And then monitoring, we're huge into tech here. We love smartwatches and activity monitors and things like that. But, and you can also look at things like pain and fatigue through whether it be journaling or some phone apps. But what we really want to talk about is what we found to be really important as we work with our patients here, and that is looking at the exertion level of the activity that you do. So we use an RPE, rating of perceived exertion scale. You can Google that. There's several different types. I happen to like the one that's just 1 to 10. But it is your impression of how hard you are working at a particular exercise. You can also do a whole session RPE, but I kind of like to be able to say, well, which exercises are really difficult for me and which ones are not. And you can, again, you just adjust these exercises up and down as needed. And then again, I think we've touched on this already, but physical performance technique, the way that you do it in a controlled manner, you can either look at yourself in a mirror, or you can have an exercise buddy or a trainer or a therapist. But when that performance starts to change, that's your body seeking other muscles and trying to get other compensatory movements to start to come in. That means your muscles are getting fatigued. We'd rather you just kind of stop the exercise at that point. Just next time, downgrade it just a little bit so that you can get those 10 perfect reps in as your strength training.
So one really important part of monitoring your progress is looking for some of these warning signs that you might be pushing your muscles too hard. So on the green side over here, we have, these are some things we would be okay with. So these are signs that you're exercising your muscle in a healthy way and we would expect to see them. So these include things like just a little tightness or aching or some mild soreness in the active muscles that you were exercising. We would also expect some soreness that can occur 1 or 2 days after. We call this delayed onset muscle soreness. And we would expect this soreness to get better as you move around and you stretch. Over on the red side, these are some signs that you might be pushing your muscles too hard. The first one is a decreased physical performance, which I want to say this is not decreased physical performance from what you could do years ago. This is you're suddenly unable to do what you were just doing last week. So that could be a sign that you're pushing yourself too hard. Some others are sharp muscle or joint pain during or after exercising, and this pain does not get better with moving or stretching. We also don't want soreness lasting more than 3 or 4 days. We don't want to see more frequent sickness, and also look out for increased malaise, depression, or anxiety. So I want to say everything on this slide has been researched in healthy muscle. So this is not specific to people with KD, but we do want to make you aware of all of this because these are things you can look out for yourself as you are starting this exercise program.
In summary, we just basically want to say that movement, we like to call it healthy movement, nutritious movement, but any movement is better than no movement when it comes to improving the health and the quality of life that you may have. We haven't put a plug in for water walking and some other type of supportive type exercises like a recumbent cycle and things like that. But these are great ways to get started where you're at much less risk of injury, but you have a much higher ability to enjoy that activity. Take inventory of your current health status and abilities with your care provider before beginning a new exercise program. Exercise in a way that makes you happy and helps you achieve the goals that you and your practitioner are setting, and then monitor and adjust as needed. We are gonna just stop here. We got some resources, and you'll see on the Kennedy's, it's a little bit of a jog there. Kennedy's, then Living with KD, then additional resources, videos, Joe Shrader. But you can navigate there, and I think ultimately it points you to NIH YouTube. But we looked at these before the conference and reviewed them all, and we like them, but we still think there's 1 or 2 that are short. So we're gonna get back to it and try to make a couple of additional videos. One in particular is sort of caregiver assisted exercises that will be safe for both the caregiver and the patient. We want to acknowledge Dr. Joe, who works in rehab medicine, who just does a ton of work with these men with KD. And of course, none of this would, for us in rehab medicine, would be possible without the whole team from NINDS, Angela Kokenius, Dr. Apatani, Grant Syke, and Fishbeck. We really enjoy working with all you guys, but of utmost importance is the patients that come in. We genuinely really like every patient that has come in with KD, and we're just so thrilled to be a part of the research and learn from you, and then pass that on to other patients. I just feel a deep sense of connection with you guys. If there are time for questions, we could take some questions, but I don't know, Terry, if there is.
I don't see any questions in the room. Yes, we have one back here. 1 second, Joe.
Okay.
I have just a couple minutes for questions.
Hi, just really quickly, I'm familiar with a really small study out of Denmark where Kennedy's disease patients, I think it was 5 healthy and 5 Kennedy's disease patients, actually performed high intensity interval training, cycling. And the thought was that you would get maximum benefit with hardly any damage to the muscles. And I think they found that increased VO max occurred in all the patients, increased workload, and there were no increases in creatine kinase or perceived fatigue. And also the patients just liked it better. So I wondered if that was something, this was really small. Is it something that you've recommended to your patients or had any success with? Do you think it would be a good idea, bad idea? Again, this was a small pilot study.
Yeah, I mean, that's the study that we love. It's in one of the slides that we presented. It's the 2019 Hedgie paper. You described it beautifully. It is a small study, so we really would like to see more larger numbers and things like that. There were 5 and 5, but 2 dropped out of one of each group, so it was really a 4 and 4. But the bottom line is everything you said was true. And I think they also, in terms of preserving muscle and not damaging it, they also felt like there wasn't as much neuronal fatigue because you were working for such shorter periods of time. That was one of their big take homes. The only, I mean, we would love to do similar types of work like that. The only thing I'll say is that the second case study that just came out, I believe in 2022, we kind of tried to model our exercise program, which was which were 30-second intervals of 15 different exercises. And there was around 30 seconds of work and then roughly 30 seconds to a minute of rest. So he, that gentleman was working very hard. That's the closest thing that we have where we have experience with doing that. We don't quite feel that we can just blanketly, give a recommendation, hey, try high intensity interval training. That specific protocol was pretty neat too, because if you look into it, it's sort of 10 seconds of high, all out work. It was 30 seconds and 20 seconds and 10 seconds of sort of low and then medium and then high. So we would just be careful about someone going to like a high intensity training class that may not be using that specific type of approach. But anyway, thank you. I think that's the most positive research that we can see at this point.
Joe, we had one more question online. What about daily stretching?
So daily stretching was the control group in our 2015 study. And we assumed, because when we look at our patients with KD, we do not really see joint contractures as part of this disease. And so we thought that the stretching would really act as a control group. And actually the stretching group also showed some gains. So we were a little surprised, but also happy by that. A lot of folks have reported that a nice stretching program, has been beneficial to them. So we do include stretching with every single patient that we see where we make a program. There's going to be some flexibility work inserted into that program, and we think that's a high value activity.
Joe, thank you so much. Another great presentation. You've put a lot of information in a very short time, and I'm sure we got a lot out of it here, and people will go back and review your talk and your slides and your videos after the conference. I'm sure a lot of valuable information. So thank you.
Pleasure.
Thank you.
Good luck, everybody.